Well, thank you to the organizers, and thanks for coming. So I'm going to just build on this background that we've heard, because the sickle cell approach is to take bone marrow, which after all is an organ in suspension, so we can get a hold of it, modify it, and then reinfuse it. So that's what gene therapy for immune deficiencies also involves at this point. And I'm going to give a little background and then focus on a founder mutation that causes frequent severe combined immunodeficiency in Navajo. And just as an introduction, what's different about a severe immune deficiency from the other things we've been hearing about is that if you don't have an immune system, then we don't have to worry about chemotherapy or the inflammation, the body's attack on the gene therapy or incoming cells, because the severe immune deficiency renders the patient incapable of doing that. But unfortunately, babies with this kind of immune deficiency have no T lymphocytes and no antibodies, so they're unable to fight infections at all. They develop recurrent infections and start to lose weight from the age of 2 to 4 months, and they get opportunistic infections with organisms that wouldn't make you or me sick at all. And so these infants can't survive unless we can do something to give them an immune system. And the very first treatment was bone marrow transplant. We've already heard a matched, HLA-matched sibling can be a donor. And this was first accomplished in the immune deficiencies in SCID, which is really a pioneer disease. So in 1968 was the first successful bone marrow transplant in humans for and it was in a patient with SCID who got a transplant from his sister, and now he's a 52-year-old accountant, and he's perfectly healthy and totally cured, and he's a dad. So we can do transplants not only from matched siblings, but also alternative donors. And in a particular genotype of SCID, due to adenosine deaminase deficiency, there's enzyme replacement that has been used as a treatment. And all these have been successive sort of scientific advances, as well as helping patients survive along the way. And gene therapy also was developed for ADA SCID and X-linked SCID, and the treatment of these conditions was the first use of gene therapy in humans that was considered curative. So we're dealing with many different genes and also different sort of personal histories. And so here is just shown data, and this is very important, the sort of preclinical registry data from the Primary Immune Deficiency Treatment Consortium, which is a rare disease consortium supported by NIH, both NIAID and the Office of Rare Diseases. And so this is a collection of patients from 43 centers around the country, and you can see that early age of transplant is associated with best survival, and patients who have infections do very poorly, even with modern supportive care. And so this led to the idea that gene therapy could be, newborn screening for SCID could be important to identify patients early before they got sick. So just to focus on the Navajo and Apache Indians, who are actually related to each other, they carry a recessive SCID mutation in the gene called Artemis, and this was present in survivors of the Long Walk, which was in 1864 when Indians were rounded up and marched from Arizona to a real hellhole in New Mexico. About 90% of them died, and the ensuing population bottleneck is thought to have increased the population incidence of this recessive gene, which was described by Hayward and Hu in 1980 when they found 1 in 2,000 Navajo infants had SCID. And the Artemis protein functions in VDJ recombination, which is necessary for T cell and B cell development, but it's also a DNA repair enzyme, and so it confers in every cell of the body sensitivity to radiation that makes these patients experience high toxicity if they're given chemotherapy, for example, in the context of a transplant. And so this is just Mort Cowan's experience with, I think this is 17 or 20 patients who he has treated with transplant at UCSF over a period of 24 years. And there are some patients who had a affected sibling, and so they were recognized right at birth. And those have much better survival than the sporadic cases who come to attention only because of their infections. And this is mirrored in the rest of the population, not just Navajos. So it turns out that there is a biomarker called T cell receptor excision circles. And this is a byproduct of the T cell receptor recombination that goes on when T cells are maturing. And these circles, or TRECs, can be measured in the dried blood spots that babies are getting for newborn screening. And so it has become a newborn screening test for SCID. And early on, I did, with many collaborators, a Navajo screening study on 2 reservation hospitals. And this was quite a difficult thing because the Navajo Nation tribal IRB had prohibited any genetic testing in I think it was 2003 that this prohibition was made because of the exploitation of Navajos and other Indians by population geneticists, and people who went in to study them claimed they were studying one thing, like diabetes, ended up studying another, like schizophrenia, without consent. And so needless to say, there's a lot of distrust among Native Americans. So we were required to have a written face-to-face consent for the babies who were enrolled. They got an extra drop of blood on a newborn screening card that was tested for these TRECs. And so we did a lot of education and introduction, both in the population and in OB clinics and among doctors. So we had this poster that said, "Silly rabbit, TRECs are for SCIDs." And then we also had the consent form with a pamphlet, an information pamphlet. And here it was very important to use appropriate kinds of language. So Native Americans often think if you say, well, you could have this or you could have that, just by bringing it up, you are causing something to happen. So if you say your baby could have this, then parents may think, okay, now you've cursed my baby and my baby's going to have that. So these are written. Some babies get these kinds of problems. We're not saying your baby. Okay, so in 2009, that yellow rectangle is the Navajo reservation. We started screening there, and you can see at that time screening was not common. It was just starting in other states. And we did get the PerkinElmer company to donate 6,000 tests to get the rest of the reservation screened. So the Navajos had this as standard of care very early. Now, of course, every state has adopted newborn screening for SCID, and this is, again, that PIDTC data that shows increasing diagnosis of SCID by screening and decreasing diagnosis by infection from 2010 to 16. And of course, now every baby in the US is being screened. So you'd think, well, problem solved, but actually not, because you're never done. Things are never really solved. But one thing that has become clear is that genotype matters. And so there are many different SCID genes. This is showing survival of patients in that PIDTC registry according to their genotype. And this Artemis gene has the worst survival, significantly worse than other genotypes. And that's probably because of this radiation sensitivity that patients have, as well as the, there are other aspects of it too. It's a T minus, B minus, NK plus SCID, and the natural killer cells that are present are somewhat activated, and so they tend to make the patients resist grafts. So even with a matched sibling, it's a very difficult disease to treat, with often poor T cell recovery and very rarely B cell recovery. So these patients need antibody infusions for the rest of their life. And these things combined to make us think about gene therapy specifically for Artemis SCID as a potentially better approach. And so with Scott McIver at UMN, we developed this vector, and it is one of the first, if not the first in human, where the natural promoter for the Artemis gene is what we're using to drive expression of the gene. And so this, again, is, as we heard about with sickle cell, it's a self-inactivating lentiviral vector based on HIV. We're using a very, very low dose of busulfan to make space in the bone marrow because studies in X-linked and ADA-deficient gene therapy have shown that without any making space at all, you don't get multi-lineage engraftment. So we felt we needed a little bit, but again, our trial has the lowest dose of busulfan that's ever been used in humans. And we're enrolling both newly diagnosed infants and previously transplanted children and adults with poor immunity in this trial. And we sort of figure if a baby comes along, we'll stop everything and treat the baby as an emergency. And then in between, we'll enroll these older individuals. So we opened the trial in 2018, and by now we've treated 4 infants and 3 older patients. Okay. And I just want to say newborn screening has been critical to this because that gives us unbiased population-level ability to make a diagnosis early. And so this promotes fair access to the treatment, because before, you can imagine patients living in this kind of territory would not be likely to get to a doctor or a medical center, let alone a tertiary care center. So how do we go about enrolling people? Well, we have found that Navajos of course, they love their kids, they want the best for their children, and even if they may not be school educated, they understand very deeply about risks, and I think about how early treatment is better. Definitely our outcomes are challenged by their environment and their poverty. We try to deal with them, and I couldn't put in a picture of our actual patients, so I have one from the archives. Our patients bring their cradle boards from the reservation, which are all wiped down with Clorox before they go into the baby's room. And we do our best to try to help the culture shock that is definitely involved. And of course, the babies have to stay in these isolation rooms until T cells develop, which takes 3 months. And so it's a long time away from home. And I'll stop there with many thanks to many people, especially to CIRM, who is funding the clinical trial that I just talked about.